bioinformatics.
Okay, and welcome to Genomics, Proteomics, and Bioinformatics, or Bio224. I'm Clayton Visger, and this is going to be the very first lecture that we have. So, this class, I think, lends itself heavily towards the bioinformatics side of things. And so, first I want to get out of the way, what is bioinformatics? And there's not really an easy answer, because if you asked 10 bioinformaticians, you'd get 10 different answers. What I like to define bioinformatics as, and many other people do as well, is it's really a discipline-specific form of data science. Data science is kind of a hot thing that you may have heard about, which is really working with huge data sets using both kind of a combination of computer science and statistics. So we have a little Venn diagram here of what data science is. Bioinformatics is really combining data science with a discipline-specific interest. In the case of this, it's biology. And so bioinformatics is really the intersection of those. You could think about just computer science and biology, the intersection being computational biology, and bio and stats being biostatistics. Now for this class, we can't cover every single aspect of everything. And stats are very subdiscipline specific, so I don't get into a lot of stats in this class. We use some stats as they apply to different analyses and programs that we're going to be learning, but we're not going to dive deep onto them. Instead, I focus most of this class in giving you enough of this kind of computational biology skill set to be able to start applying bioinformatics at an intermediate level, and specifically getting yourself to the point where you can start to learn it. Okay, the syllabus, you should have all access to it on Canvas. It's got kind of the typical stuff. I'm not going to go into crazy depth here. Most importantly, if you ever forget, I have the links to the Zoom for both my office hours, which will be 2 to 3:30 Monday, Wednesday, so after this class, well, a little while after this class, as well as where the Zoom link for our reoccurring lecture/lab period will be. Now, go ahead and read through this kind of on your own. But you can read through kind of the course description, and we'll talk more about that as we go, as well as the software that are required to be installed. You'll be doing some of that, getting kind of the bare minimum installed and up and running this week as a part of the first lab. So don't worry too much about that. And then I've got some recommended resources here, things for as we get a little further along in the class. If you start to get stuck as far as coding is concerned, there are some great resources. Google is, there's nothing wrong with Googling things, but there's also BioStars and Stack Exchange, as well as GitHub, that are nice resources. The objectives for the course really are to gain a basic set of computational skills and understand how to apply those skills towards running bioinformatic analyses with large-scale biological data. In this case, we'll be using, throughout the class, genetic or genomic data, but there's nothing to stop you from applying these same principles to analyzing large-scale ecological data or anything else for that matter. Most importantly, I want you to develop the ability and, more importantly, confidence to know that, you can do this. And I want you to be able to start facilitating your own learning. And what I mean by that is, if you read in a paper, oh, that was a cool analysis that just came out, I would like to run that on my data. Well, being able to know how to figure out how to teach yourself to apply that is really how a lot of this work is done, and I want to facilitate that and develop that skill set in you, and much of this course is based around you developing that skill set. And then finally, I want you to be able to use the skills that I teach you specifically to analyze some publicly available genome data and do some basic analyses. General expectations, these are very similar to most classes. Attendance, when things are required, try to be there on time. There's always exceptional circumstances. I totally understand that, particularly now. So just as a rule of thumb, if you think there's going to be problems, just let me know and I will happily work with you regardless of what the problem is. Respect's a big one. I will do my very best to respect you guys, and I ask the same of you towards each other and towards me. It can be hard to kind of not accidentally talk over in Zoom and things like that. These are all technical issues that, are different from what I'm talking about here. Communication, just bring things up to me. If you're struggling, bring it up to me. If you have issues, bring it up to me. I am happy to help you, but I can't help you if I don't know about it. Don't cheat. This is a class where there's, not a lot of exams. We have 2 practicals. I'll be honest, if you really wanted to be texting your friend all the code that you're doing to run it and cheat, you probably could. And there's very little I could do about it. It's kind of, silly to say, but, you're really cheating yourself, that old chestnut. But I really do mean it for this class. This is not a class where I am trying to get you guys and fail you guys. If you struggle on a practical, but you did it honestly, you're going to be okay in this class. I'm not worried about that. I would much rather you struggle than pull one over on me and get an A on a practical when really you're going to be fine in the long term either way. So be honest with yourself, because it helps me figure out who is struggling in the class and who I can help. Finally, patience. This is, again, I bring this up all the time, one of the biggest things. Bioinformatics is a rapidly developing discipline. We're going to run into all sorts of issues. I try to troubleshoot way ahead of time. Every single semester, I'm troubleshooting these labs, and I get through them with no issues. And then all of a sudden, some weird bug will pop up the day of, and multiple people have the same issue, and we got to figure out what's causing it. And it could be all sorts of things. Many of them are not your fault. Some of them are my fault. Some of them are no one's fault. But they're all opportunities just to get better at dealing with troubleshooting, which is a big part of this. All right, grades. You can look at what these different assignments are. In a nutshell, I've added a couple assignments this semester because we're not around each other. And so I kind of wanted to facilitate communication a little better. So the things I've really added are these lab reflections and discussions. And we'll talk more about them as you get your first one at the end of this week. But in a nutshell, it's really just thinking about what you're doing and communicating it back to me in the case of lab reflections. And the discussions are just kind of nice, I think, semi-lighthearted ways of just kind of discussing the material or bioinformatics in general and getting to know me and your classmates a little bit better. Well, also, on the bright side, getting a lot of points that will hopefully make you feel at ease in this class. In the past, there have not been this many points in the class. It's typically been out of, I think, 200, and around 150 of those points were wrapped up in the software tutorial that was at the very end of the semester, and it caused a lot of anxiety, even though I knew you were all going to be okay. A lot of the previous students were very stressed out about that. So I've tried to add a lot of extra points in here as a buffer to kind of help make you feel a little more confident as we go, and hopefully facilitate some interesting discussion and reflection along the way. I also grade on just a standard 80, 90, 70 point scale, but I do understand that for the sake of you being master's students, a B- is considered failing. So I do not think of the average grade being a 70, anything like that. So understand that I understand the scale is that a B- is an F effectively. So just want to make sure you feel good about that. Related to our current situation, we've got some COVID-related resources here. If you feel that you may have it, you can contact and get tested and do some other stuff. So that's there in the syllabus if you need that, as well as some other resources as well, counseling, and CARES resources specific to a lot of this remote transition and basic needs for that. And then on the very end, I've got both a little breakdown of what our main project is towards the end of the semester and our schedule, which is a little zoomed in right now, but I can probably zoom in on it right here. And so here's just our general schedule. This class meets for lecture and lab twice a week. They're pretty much next to each other as far as time, so I tend to treat them as one large block of time. Most of this, I would say 90% of the time that you're in this class, it is self-paced. You're working through kind of the labs that I've set up for you. You're welcome to drop in and drop out and take breaks. I do strongly, strongly encourage you to think of these as being particularly the beginning as being important to be there synchronously, because I'll be introducing the lab or the lecture, and I'm going to be there the entire period to help you through the lab, because a lot of this is you working through the labs on your own, and you're going to have lots of questions that you may be able to answer yourself, or your lab mates may be able to answer for you, but at a certain point you're going to need to come to me for some things from time to time, and the time that I'm there for you for this is just like a normal semester in this class is during that lab period. So it's really important that you try not to put these labs off and you work on them during the designated lab period time, because that's when I have time in my schedule to help you with them as well. So typically be a lecture and then an affiliated lab back to back. So if we talk about Unix shell, we're going to have a shell tutorial as the lab for that day. If we talk about sequencing platforms, we're going to talk about sequence data. If we talk about short read alignment, we're going to practice aligning and mapping reads. What you'll notice towards the end of the semester, I actually devote a lot of time towards open labs where you're working on your software tutorial project and I'm helping you guys with that, answering questions, troubleshooting with you. On the right, we've got all the assignments. I've got a couple reading assignments peppered throughout the semester, but there's not a lot of homework in this class. The closest thing to homework that we have is we're going to have that weekly lab reflection, and there's going to be 7 discussions throughout the semester, as well as 1 or 2 little tiny mini assignments that are kind of shouldn't take you more than an hour or 2 max. So really the bulk of the at-home work for this class is this kind of major project. And that won't really be something that we start dealing with for at least a couple weeks, maybe a month, until you start thinking about what your focus is going to be. So one thing I'd like to do is, very first day of this class, we don't talk a lot about the biology of things after today. And so I expect most of you come in with a solid background in genetics and molecular biology, given your biology master's students, and this is a bioinformatics class. But I thought I would just touch on a few things just to kind of make sure we're all on the same page. Hopefully everyone here is aware of the central dogma of molecular biology, this concept of DNA to RNA to protein. That's very, very important because we look at data sets related to each level of the central dogma. And so we look at, DNA, and we can think of all the DNA in a cell as being the genome. We can think of all the RNA or transcript in a cell as being the transcriptome. We can think of all the protein in a cell as being the proteome, or all the metabolites as being the metabolome, or all the phenotypes of an organism, a whole species, all the sizes and variations in color as being the phenome. And so bioinformatics really deals with these large kind of views of these things, looking at the genome and patterns within it, looking at the transcriptome and patterns within it, not so much at a single gene or a single transcript. Now, I said ohm a lot. If you're curious, ohm is actually a Greek term that tends to mean the totality of something. If you are looking at all of the genes, you're looking at the genome, although really the genome also includes non-gene regions as well, but that's kind of so you can get it a little bit. Okay, so let's walk through a couple of these things. DNA, good old review here. 4 nucleotides, A, T's, G's, and C's. That's how they pair. You can get substitutions between them, mistakes. You can get transitions where you have a mistake that replaces a purine with the other purine, so an A to a G or a C to a T. You can have a transversion where you have a purine or a pyrimidine being substituted with the other type. This could be 4 possibilities: C to G, T to A, C to A, or T to G. Transitions are much more likely to occur than transversions. And so why does this relate to bioinformatics? Because as we're starting to look at whole genomes, we need to start thinking about ways we can look for patterns and pieces of information. One piece of information that's used quite a bit is the estimated rate of base pair substitutions, particularly as it relates to phylogenetics. So in phylogenetics, we're often looking at different sequences from the same gene between different species and trying to estimate, based on the differences between them, which was most likely to have diverged the most recently, and so on and so forth. And there's different models of base substitution that we can use. We could use very basic JC models where they're all the same. There's no difference between the frequency of transitions and transversions, all the way up to a model where every single base substitution combination has a slightly different rate that has been estimated. Now, all these base pairs occur typically in either what we think of as kind of genes or non-gene regions. And so the basic makeup of a eukaryotic gene, if you're not familiar or need a refresher, we typically have kind of a coding area, and then we've got some regulatory sequences both upstream and downstream. We're not going to talk much about that stuff. The reason I mention this is because as it pertains to this class, this is where the relationship between the genome and the transcriptome is. The genome is going to have all of this material, whereas the transcriptome is just going to have that translated RNA, transcribed RNA. And this can be informative also, because eukaryotes, the structure of these have a 5' cap and a poly A tail, and you'll see later, or already probably know, that prokaryotes do not. So again, here's that DNA to RNA structure. We've got that 5' cap, we've got bases, we've got a poly A tail on the end of these. Why do I mention this? Well, let's say you're sequencing a bunch of RNA and you are worried that you had some contamination in the lab. There's a microbiologist that works, at the same bench as you, and they spilled a bunch of bacteria the day before and forgot to tell you. It'd be very easy to sift through all your RNA transcript sequence and pull out anything that doesn't have a poly A tail, right? If it doesn't have a poly A tail, it's probably bacterial, and you could throw it away. So that's one way some of the basic biology can be informative. Next, these transcripts are then translated, right, by ribosomes, and most of you or all of you should be familiar with this, all the kind of concept of codons and the wobble between them, yet there's a lot of redundancy, particularly as it relates to the third base. So a CCC is the same as a CCA as far as which amino acid it codes for. And so, again, going back to thinking about patterns in the genome, you would expect a base pair substitution that does not change the amino acid, or a silent mutation basically, to be much more frequent in the evolutionary history of an organism than one that changes it from a amino acid to another amino acid. If they're of the same polarity, you would expect that to be more likely, though, than if it was to change to an amino acid with a different polarity, or if it disrupted a sulfide bridge, something like that. So all of these kind of basic concepts inform us when we think about what's more likely from kind of a frequency perspective. Okay, and then here's just a breakdown of kind of how we term some of these different mutations or substitutions. You can have silent, where it doesn't change the amino acid, nonsense, where it inserts a stop codon, and missense, where it changes the amino acid. Here's just a nice figure illustrating, again, that DNA is double-stranded. You generate your RNA transcript from one side. The side that you pair against when you're making, that you complement against when you are making that RNA transcript, is what we call the template side. It forms a template for reconstructing the coding strand. Your RNA is not generated or coded from the coding strand. Your RNA reflects the coding strand because that's what's ultimately producing the amino acid code. So that's just kind of something that might come back up a little bit later in this class. Once you've got all your amino acids, right, you've got different levels of structure. The primary structure is the sequence of the amino acids. You've got your secondary structure, whether it's squiggly or foldy. You've got your tertiary structure, how that whole thing is kind of crumpled up. And then your quaternary protein structure is when you have multiple amino acid chains that are joining together, like a hemoglobin, for example. So why did I go through all this? Because it's important to think about how much every single one of these steps can impact the ultimate phenotype of an organism. Typically, we always think about, change occurs at the DNA level. There's mutation, things like that. But really, you can think about mutation being a driver of phenotypic change. Alterations in the regulation of RNA, when it's being transcribed or not, whether it's daytime or nighttime, that can impact the phenotype or behavior of an organism. How that RNA is spliced, how that RNA is modified after translation, how that amino acid string is being localized to areas of the cell or beyond. And then epigenetic mechanisms, what's changing in methylation patterns, things like that. Now, these are all things that we can look at from a bioinformatic perspective in one way or another. And these are all steps or stages at which change can be realized at the phenotype. So let's talk about how a couple of these big things actually vary between some major groups, particularly eukaryotes and prokaryotes. So the genome, again, is housed within chromosomes. In the case of prokaryotes, it's one large circular chromosome with potentially extra genes being harbored on small circular plasmids. The prokaryote genome, contrary to the eukaryote genome, is on by default. If you don't do anything to this genome, it's going to express all its genes. Repressors have to actively stop expression. There's also generally no introns or no alternative splicing and no poly-A tails on their RNA. Now, there are exceptions to these. There are some examples of prokaryote introns and things like that. But in general, the genes are on unless you tell them to turn off, and there's not a lot of funny business with the RNA. The eukaryote genome is actually linear chromosomes, and it can be as few as 4 to as many as over 1,000 chromosomes per cell in some fern species. So quite variable. It is typically off with respect to expression, and so you need to actively turn it on through a number of different regulatory mechanisms. Alternative splicing is rampant, as well as the use of alternative splicing to skip exons or even include introns. When we think about the distance across genomes, particularly linear chromosomes, we can really think about them being measured in 2 ways. Physically, how many base pairs are between this point and this point? 1,000, 10,000, 1,000,000, 1,000,000,000. That's a physical distance. Now, it's not a measured distance because the actual distance as far as micrometers varies because chromosomes condense and uncondense constantly. But if you were to unwind it into one chain of double-stranded DNA, you would be able to count the number of bases, and that would be consistent between 2 regions. So that's the physical distance. Now, hopefully you all remember that during meiosis, there's this thing called crossover, right? And the genetic distance is measured in centimorgans. And that's really the distance based on the frequency of recombination and how much that differs from the expected 50%. Now that will vary. What 30 centimorgans is represented on the chromosome will vary as you move it around because there are regions of the chromosome that have different rates of recombination as well. And so recombination, while we're talking about it, let's talk about some of the wacky things chromosomes can do. Let's say we've got these example chromosomes with 8 genes on them. You can have deletions of whole genes. You can have duplications of genes and tandem duplications. So you can go from having 2, 3, 4, 5 to 2, 3, 2, 3, 4, 5. You can have inversions where it goes 2, 3, 4 to 4, 3, 2. You can have translocations where you accidentally end up with a crossover between 2 different types of chromosomes. And you can have it be reciprocal, where they each keep the respective half. You can also have it non-reciprocal, where effectively one side is converted to the other side. And then you can have transpositions, where one piece moves all the way around to another area within the chromosome. On the subject of duplications within chromosomes, particularly gene duplications, I like to think a lot about what happens. What are the fates of duplicate genes? If you have one gene and now you have 2, well, you've got a lot of redundancy, right? You've got 2 genes that do the same thing. What's evolution going to do to that? Well, it could tolerate one of them being broken. Maybe it gets a nonsense mutation. But it's not going to functionally impact because you still have one good copy. So non-functionalization of one copy is pretty common. It might be that if you doubled a bunch of stuff, that you might need to conserve for the sake of stoichiometry both copies. And so you could end up with conservation of both copies. Just like having a nonsense mutation, you could by chance get a mutation that is actually beneficial and serves a slightly different role, where now this copy is doing something it never did or is expressed in a tissue it never was previously because it's free to move into that area because the other one's got its original use covered. And then finally, my favorite is subfunctionalization, where you basically, each copy ends up specializing on one subset of the original whole role of the gene. So, real simple example, think of day and nighttime. If you've got some gene that's typically expressed 24 hours a day, well, if you subfunctionalize 2 copies of it, it might be on in one copy and specialized for daytime use, and the other copy might specialize for nighttime use. This is often what we see happening to many members of gene families. This is something that we saw happen with the Hox genes, for example, if you're familiar with those. And so this is all very cool, the fates of duplicate genes. And what really interests me a lot, just to give you a background on my interests, are when you duplicate all the genes. So whole genome duplication or polyploidy is a specialty of mine. And there's two types. There is allopolyploidy, where you start with two diploid species, separate species. This one's got a different number of chromosomes than the other. And if they happen to make a mistake during meiosis and produce diploid gametes instead of haploid gametes, which actually happens fairly frequently, around 1 to 2% of all gametes are actually unreduced. They're diploid gametes. So if you have 1 to 2% happening all the time, it's only inevitable for those 1 to 2% diploid gametes to meet and fuse and make what we would call an allotetraploid. This is a cell or a zygote that has the entire genome of two different species in it with its own set of paired homologs. This is cool because you've got preferential pairing. Each of these has a partner to dance with, but they also have, because often these species, these two diploid species are closely related, these are often fairly close in what kind of genes are located on them. So it's almost like having slightly divergent genome duplications. And in most cases, there will be preferential pairing. The blue will only pair with the blue, but sometimes you can get translocations between these things. But what's cool is you're basically forever heterozygous, because whatever gene is right here on this red one could become homozygous here. It probably will very quickly, especially if you're in a small population size. But the copy from the other species here probably serves a similar role if they're not that divergent, but it will never recombine for the most part here. So you're effectively forever going to have at least two different copies or two different alleles for every gene in the genome. And this kind of fixed heterozygosity genome-wide often leads to some pretty interesting things. A lot of my kind of previous research experience, though, is specializing on autopolyploidy, which is one species that doubles everything. And so you go from having 2 of every chromosome to 4 of every chromosome, or 8, or 12, or 16. What's cool about this is all these copies pair at random, so you end up with weird inheritance patterns, but you also end up with strange gene dosage issues, where you could have a lot of upregulation in some genes, forcing the upregulation of other genes that will bring them down, and thinking about what happens to the expression of these genes across the genome is something that really interests me. I mention this because although we don't think about it a lot as being a contemporary issue for humans, polyploidy is rampant across the eukaryotic tree of life. The ancestry of major lineages of animals have many ancestral genome duplications, as well as plants, and many of these organisms are having them constantly pop up in populations as well, and are, at least in plants, responsible for up to 70% of estimated speciation events. So really important in some major lineages. Okay, so that was just kind of a quick walk through some biology that I think might be relevant in this class, but we'll go ahead and move on to why might we need bioinformatics? So way back in the day when computers were rapidly becoming more powerful, and they still are, there was this idea of Moore's law that was developed, which was based on the rate of transistor counts per microprocessor and the rate of increase in our capacity to produce those. And really what Moore's law says is that every 18 months, our capacity for transistors per microprocessor doubled. In other words, our computing power doubled every 18 months. And you can see here, this is a nice linear growth. If we look at the cost to sequence a raw megabase of DNA, starting in 2001 to about 2007, it follows the expectations of Moore's law, meaning the decrease in cost of sequencing basically was halved, or every 18 months, arguably with kind of following along with our increase in microprocessing. So we were always able to keep pace with our computer power and our generation of data. So something changed though. Starting in 2007, the advent and popularization of next-gen sequencing technologies, really high throughput sequencing, put this off at kind of an exponential growth of the amount of sequence data you could get for your dollar. And so all of a sudden we were under this deluge of sequence data that didn't keep pace with our capacity to process it computationally. And so this is where I think bioinformatics comes in and becomes really important and popular. How do we deal with the disparity between our computational growth and our data growth? So let's give you an example of how much data we're really talking about. So genomes, they vary tremendously in size. Viruses can be on the order of kb, or thousands of base pairs. Bacteria are on the orders of mega base pairs, 1 million base pairs or so. And then eukaryotes can be, a gigabase, 1 billion base pairs. That's a relatively small genome, a very moderate genome. You can see we have plants go up to 100 gigabases, much, much larger. And some protists get really large. So these vary tremendously in size, but for the sake of argument, let's say you just want to sequence an average eukaryote of a 1 gigabase genome. Typically, and we'll talk about genome assembly, you want around 50 times as much sequencing as is the size of your genome. We call that coverage. So if you have a 1 gigabase or 1 billion base pair long genome, you want about 50 billion characters of sequenced data. But double that number because every base pair has a quality score when you first get it from the sequencer. So we're looking at a file that has 100 billion characters of data. What does 100 billion characters of raw information even look like? I've never seen 1 billion of something, let alone 1 million of something, so it's hard for our brains to even process how much data that really is. And so I like to use this example that the world's tallest Lego tower is 126 feet tall and is made of 500,000 Legos. That is a lot of Legos, and that's a big tower. It can't even support its own weight without all those cables. Imagine if each one of those Legos represented one piece of data, one base pair, one letter. It would take 200,000 of those towers to have enough data to build a 1 gigabase genome of mediocre quality. And we're generating way more data than that on a daily basis. So there's really no way to look physically at your data. It's too large. We need to look at statistics, and we need to look at analyses and the output of those analyses to try and gain an idea about patterns within our data. And this is one of my favorite quotes from a popular bioinformatician, Aaron Quinlan. Analyzing your data means inherently distrusting your data until you've exhausted yourself into giving up and trusting it. So kind of the take home here is that the volume of genomic data is really hard to comprehend. I showed you an example of a small set. And so that makes it very difficult to even think about how to look at it, much less how to analyze it. And so that's where bioinformatics comes in. That is the tool that we use to handle biology's really big data set, genome sequence data, and how we analyze it. We're data scientists with respect to genome data in many cases. So I'd like to give you an example that kind of came to me when I was my first semester here, when I was also simultaneously planning on getting married and was thinking about wedding invites. Some of you may have been through this already. You end up with this Excel file, and you need to send out invites, and you've got all the people's names and addresses, and how do you get those on envelopes? Well, there's two options. You could go through your Excel sheet and type out the label and print it or whatever for every single person individually. The amount of time that's going to take is dependent entirely on how many people you're going to invite. It's X amount of time per invite. So maybe it takes you 2 minutes. If there's 10 people, it takes you 20 minutes. That's well and good if you're starting a small wedding with 10, 15 invitees. What if you're famous and you're going to invite 5,000? Is that something you want to do by hand? So an alternative is that you could write a script that would pull all the names out from one column and the addresses from another and reformat them into a format that your label printer will use. And so the amount of time it would take to print all those invite labels is Y, a fixed time regardless of invites, because the time is all soaked up into writing the script. And then once you have it written, you just press run and it doesn't matter how many invites you take. And so there's this decision we always have to make when we're doing these types of things, is how much time would it take me on a fixed per task time time frame, like the handwriting, the more things you need to do, the more time it's going to be spent. It's a linear relationship. Whereas maybe you start doing that. You do a couple of them and then you say, screw it, that's way too many. You stop doing them and then you spend all this time writing this script. And you write this script and you write this script and you're not making any more progress. And this other person in that same amount of time is laughing at you because they're getting closer to being done and they've passed you. But then you run your script and there's no more time spent. You're done. And so figuring out where this break point is between where automating it wins out over doing it by hand is something we always have to think about. The one thing I think that's important to remember is every time you write scripts to automate things, you learn a lot also. So I find myself always doing things the harder way because it forces me to continue to learn and get better at what I'm doing. So don't always think about, oh, what we're doing right now is really inefficient, because it might be inefficient in just that one task, but it's an efficient way to teach yourself as well. So we're often writing these scripts and then not deploying them on our computers because our computers are not very powerful relative to the amount of data we need to look at. Research computing is something that's huge in both academia and industry now. There's large server farms where you actually are sending, you're interfacing directly with the computers that are in these servers and having them run your analyses. Or really, it's you running it, but on that architecture. And so in this class, we're actually going to be having all of you make your own servers, small servers, but it's still the exact way I would interface them back when I was working with HyperGator at University of Florida, where you're sitting here on your little computer and you're typing in the terminal. And what you type in the terminal is not actually interacting with your computer. It's interacting with a computer or multiple computers together in a remote server. And so that's what we're going to be doing. We're going to be using specifically the shell language and a little bit of R to communicate with our remote servers, because most of these servers, they're not running Windows. They're typically running Linux or another flavor of Unix like Ubuntu. And you're not interfacing with them in a graphical way. So we're going to be doing everything in a text kind of environment, a command line environment, because that's how these things are done in the real world. And so we'll focus on developing some nice Unix or Linux shell and bash coding skills, as well as some R a little bit towards the end of the semester for some downstream analyses. I feel it's important to point out, again, patience, but don't get frustrated when you hit a wall. I found after teaching this a few years now that a lot of students come in expecting that people that code like you see in the movies, like this guy here. Nobody I know writes code, just sits down and starts banging out some code. You're mostly, especially from bioinformatics, where you're bringing a biology skill set that pure computer scientists don't have. You don't need to be as good as computer scientists. You need to be good enough to apply the things that you want to apply for your specific research questions. And so really, I think of myself as a researcher exploring the wealth of code knowledge to figure out how to do something. I often will write in plain English what I want to do, and then I will set about finding snippets of usable code from other projects, from other people that do subparts of all I want to do. And then my task is to try and string it together into something usable that does what I want in a way that I hope is not introducing a bunch of artifacts or error. So that's kind of the expectation of skills for you guys to develop in this class. And I'm not the only one that views it that way. If anyone's familiar with the onion, well, there's a scientific version called the allium. Allium is the genus for onion. And so they are always kind of putting out kind of funny things about how computer programming is going to be officially renamed Googling Stack Overflow, which, as you remember from the syllabus, is something I suggested you do, because even computer scientists do it. Why write a bunch of new code if someone's already written it for you? It's just a tool. You wouldn't go build your own pipetter if you needed to pipette a volume of liquid and you were in the wet lab. You would use another company or another person's pipetter. Again, copying and pasting from Stack Overflow. These are all just kind of things that make me laugh a little bit and are very true to how things are done. So I mentioned that you're going to be generating your own servers this semester. I've mentioned it in some of our announcements earlier as well, I believe. Part of the reason for this, just to give you background, is that you're all on different operating systems. You're all on different versions of the same operating systems. What works on Windows 7 won't work on Windows, whatever Windows we're on now, 95, 10, whatever. And same with different versions of the Mac OS or Ubuntu or Chrome OS. And so rather than dealing with every single different person's problems differently, which would be an impossible task, we're all going to make identical instances using resources available through Amazon's Elastic Computing network. All of you, when you sign up, will get around $100 of free credit for Elastic Compute. This should be plenty for all the labs. The only caveat is that you have to start and stop your own servers. If you leave them on, you get billed per hour. So there is the potential that you could use up your free credits. The servers I'm going to have you make are pretty much the same as a desktop computer as far as speed. And they only cost something around 10 cents an hour. So you'd have to leave it on a long time to run out of credit by the end of this class. But if for some reason you do make that mistake and you do end up coming out of pocket a little bit for computing time, one, let me know. But two, also please keep in mind that there's no textbook or anything that I require for this class. But that said, I've never had anybody go over. So just be good about stopping your servers. You'll walk out of here with still probably having $60 worth of credit. Okay, so what we're going to be doing today, or if you're watching this earlier in the first lab, is you're going to be following the videos from the playlist under Resources and Setup module. These are some videos I put together for how to sign up for Amazon's Educate, as well as how to make your first server, and then how to log into your first server and perform the initial setup, and then some directions on your password file. You'll see when you watch the videos that your password is actually a file rather than words. I've got a folder up in the Resources module as well for you to upload your file so that I keep a backup copy for you and so I can help you if I need to log into your server. And then shut down your server so you don't run out of money. And then spend the rest of the week preparing for the next lab, which there's nothing to prepare for, and skimming a paper that I've thought you might like just to get an insight into coding for biologists. So with that, welcome to the class, and as you work through the first lab, I will be there to help you.